This was turning into a really long presentation, so I've decided to go ahead and split this up into two, so you will just continue in your notes from Chapter 6, Part 2. Almost all microbes are going to live in a shared habitat in a symbiotic relationship, meaning they're going to have some kind of a long-term association between organisms, all right? So looking at symbiotic, there are three main types. You can break this down even further, but we're going to look at the three main types here. These often involve nutritional interactions, okay? So symbiosis is a term to represent a situation in which two organisms live together in a close relationship, close partnership. Symbionts are members of this symbiotic relationship, okay? So mutualism here, we're going to have both benefit, okay? And so here we're going to have one partner here when symbiont is happy, and another partner here is also happy. Both are getting something necessary. The microbe perhaps gets necessary nutrients. Many microbes are going to fall in this category, but the host can also get things like protection from other microbes. So let's say, for instance, E. coli. E. coli is going to live in the gut and get nutrients that it needs, and it is also going to take up space and residence and help prevent other microbes from overgrowing or inhabiting the species as well. There are bacteria as well that also produce things like vitamin K and other vitamins in our gut, okay? I'm trying to think of the E. coli is the one that does the vitamin K, but they can produce vitamins that we actually use. So it's good for both of us, okay? Healthy development of the immune system. All right, next we have commensalism, and this is where one is going to be helped, okay? So we're going to have one here is happy. The other is not helped, but it's also not hurt. So he really doesn't care, okay? So they didn't harm or help the other member. One is helped, all right? The last one here is going to be parasitism. And so this is where one guy here is going to get food. So think of a tick. That's a really good example of a parasite. But then that tick also makes the dog that it is inhabiting sick, especially if there's a lot. So one is helped and one is harmed, all right? Some worse than others. So microbes that make humans sick fall in the category of parasitism. So non-symbiotic relationships also exist. We can have antagonism, and that is where an association between a free living species, but this can come about because of competition. That's important when we were talking about how it's important when things like normal microbiota, those that like E. coli live in the gut, take up space and keep others from moving in. That's an antagonistic type of relationship. Antibiotics produce inhibitory compounds such as or inhibitory compounds that can make it inhospitable or destroy microbes, okay? So that's another example there. Synergism is an interrelationship between organisms that benefits them, but it's not necessary to survive, okay? So they participate in a cooperative manner that produce a result that they could not do alone. And this is going to be, our example here would be like in a biofilm. So with gum disease, dental caries, and blood infections can actually involve mixed infections of bacteria cooperatively living in a synergistic relationship. I just mentioned biofilms, and these are really kind of neat little things. They're like little communities, little communes, so to speak, of bacteria living and working together as one. There's a TED Talk link, I believe, is posted that is really interesting about biofilms. It's not required, but it's pretty cool. It talks about how biofilm bacteria communicate, okay? It's a TED Talk. I can't think of the title at the moment. But so what happens with a biofilm is you may have a pioneer type colonizer. So the first guy is going to go and attach to the surface, and then other microbes are going to attach to those bacteria or a sugary protein, a substance secreted by the colonizers, and that's going to allow them to attach and stay there. And then they're going to undergo, and this is part of the title that I was just forgetting, quorum. They're going to undergo quorum sensing, which is a way that they actually communicate, because they release this special bacteria that kind of helps them to communicate and live together as one. It calls in other bacteria. And so the cell population is going to grow because of this quorum sensing. This is what that TED Talk kind of explains. It's really pretty cool. Once attached, they are going to release more of those chemicals, and more quorum sensing is going to occur. And then eventually you're going to have a large complex community form, and it is protective in nature. Okay. So a single biofilm is a partnership among many microbial inhabitants, and it serves as a protective area from them. They can be protected like that. So when you typically see biofilms, it's going to be things in like catheters or surgeries where you have placed an implant. And so when you hear somebody going in and having to get it scraped off, it's because there is a biofilm usually there, and you have to get it physically removed in order to get down to the bottom to get all of those bacteria. Because if you think about, you've got this big community of organisms, you have this little species of bacteria under here, you can't reach them because of all these other guys. Okay. Also, sometimes these can break off and escape and go through the bloodstream, and so you will hear about infections that have spread this way as well. Okay, so they can escape and become free living again, which is very problematic. Bacteria can grow in a similar fashion to fungi by budding or hyphal formation, forming hyphae. But most bacteria are going to grow via binary fission. So the parent cell is going to enlarge, as you can see over here. It is getting larger. Chromosomes are going to be duplicated, and then they're going to move to the sides of the cell. You're going to form this little protein band in the center, and a septum is going to begin to form, and the septum is complete, then the cells are going to be divided, and you're going to have two identical cells. Basically, it's cloning. So time is needed for one complete cycle of fissioning, okay? We have one parent that will go to two daughters, and it's going to increase the population by a factor of 2 every time. And this doesn't seem like much at first, but after a while, the population becomes huge. Let's watch this really short little video right here, if this works correctly. Watch how it goes from just a couple. $5,000 billion in one day. Okay, so that is a lot. This is showing you some of these growth curves. So here we're starting as that single cell, and doubling is just a little at first, but after a while, when you start doubling, it becomes exponential, okay? So the population doubles with each new cell division or generation, all right? This can be represented as a logarithm using the exponents, all right? Or it can be with simple numbers, and that's where you would get these two different lines. Plotting the log of cells produces this straight line here to the right. That's going to show our exponential growth. Plotting arithmetically, you're going to see it's got more of a curved slope. So why do we care about the math? Well, that's because we use these population growth curves to study bacteria and to make predictions, okay? So bacteria do not, thankfully, maintain that potential growth or doubling rate endlessly, thank goodness. A number of factors can prevent a bacteria from dividing at their maximum rate. A bacteria population is going to display this predictable growth pattern over time, all right? And so you do definitely need to know these steps of this growth curve that we're going over. We can observe this population growth pattern by doing what we call a standard plate count. We can observe this population growth pattern by doing that standard plate count. You count the total number of live cells in a given period of time. So these are the steps in a standard plate count here. Put a tiny number of cells into a sterile liquid medium, incubate that for several hours, sample the broth at regular intervals, and place each sample onto a solid medium, and then count colonies. Okay, so this is colony counting. So this is one of the labs that we will do or have already done. I can't remember exactly how they, hopefully they're kind of in the same. That would be really great if they were in the same area, but that doesn't always happen with lab. So this is how we count number of colonies, and this is via a serial dilution here. And so the calculation is the number of colonies times 10, and that's converting your 0.1 milliliter sample to 1 milliliter standard times the total volume of the flask, which in this case happens to be 500 milliliters. Okay, so like I said, this is often used with a serial dilution where you will dilute each of these every time. Okay, you will take 1 milliliter of here, place it into another, and add 9 milliliters of water. You would take 1 milliliter of this after you put it on your plate, put 1 in here, add another 9 milliliters of water, and so you dilute it in a series. That's what a serial dilution is. Okay, it's not always done with this, but it does often occur. So you're placing a tiny number of cells onto a sterile liquid medium here, incubating several hours, sampling, plating, and then counting your colonies. One colony on a plate is going to be a colony forming unit. So that means that all of those bacteria came from one particular bacterium, one microbe. All right, so these guys, put a little thing back on here. These two colonies here all have grown from 2 separate bacterium. Okay, and so this would be, you could pull this out and make a pure culture with that. All right. Multiplying the number of colonies in a single sample gives you an estimate of the total population size. So you can see how bad the initial infection is. Okay, so this can be graphed for each sample in the sequence for the entire incubation period. This is used a lot in the food industry. It does have limitations. It's not extremely accurate. It can't tell you, it will sometimes, if it's very early on in an infectious state, give you a 0 quantity. But this is done a lot for quick testing for food applications. So this is what your typical growth curve would look like. Okay, you need to know this, and you need to know what these stages are. You're going to have this lag phase here where you're not seeing a lot of growth, and then that exponential growth phase here, and then we're going to have a stationary phase and then a death phase. Thank goodness for that death phase, right? So the lag phase is that flat period of growth due to newly inoculated cells that require a period of adjustment, enlargement, synthesis. The cells are not yet multiplying at that maximum rate. We're early on. We're at 1 times 2 is 2, 2 times 2 is 4. Population of cells can be sparse or dilute. Sampling may miss them in this area. And then, of course, that exponential growth is when we're getting into that logarithmic phase. The growth curve increases dramatically, okay? And that's going to continue as long as there is adequate nutrients, all right? And the environment is favorable. But if you think about fish in an aquarium, you start adding too many, there's not enough food, right? They start dying off. So you're going to get to your stationary phase where your birth and death rates basically have equaled out. So they're equal, all right? Cell division rate can be slowing down here. This can be caused by depletion of those nutrients, depletion of oxygen, excretion of organic acids and other pollutants into the growth medium that will make it inhospitable for things to live. And so then once that builds up enough, you have too many fish in the aquarium, they all have to go to the bathroom, and what happens? They build up ammonia and they start dying, okay? Same thing here with the microbes. You get enough of that built up, they start to die out, all right? And so the cells begin to die at an exponential rate, which is kind of similar to their growth phase, not exactly as dramatic, but exponentially due to those waste. And then the speed with which the death occurs depends on the resistance of the species and how toxic the conditions are. And so this is slower, a little slower than that exponential growth phase, but it is variable, all right? And then we have the viable non-culturable state, and this is where many cells in the culture are in the death phase, but some are still alive, but they're dormant. They won't grow on our culture medium, and so they're missed in colony counts. So why is this growth curve important? Okay, so the tendency here for populations to exhibit phases of rapid growth and death is important for microbial control, food microbiology, and culturing. All right. Microbes in that exponential growth phase are going to be more vulnerable to antimicrobial agents. Heat agents, things can also rapidly things like heat agents can rapidly accelerate the death phase. But once you're they can in all populations actually, but in the exponential phase particularly, all right, actively growing cells are more vulnerable to conditions that disrupt cell metabolism, okay, and binary fission. They're going to be more vulnerable due to things like inhibition and destruction. A person is also actively shedding bacteria in the early and middle stages of infection. So this is also how you know if it is going to spread from person to person. So this is where they get the rule for, say, if you've been 24 hours without fever, you are no longer actively shedding bacteria. So you're less likely to give it to someone else. We can analyze bacteria without actually growing them. One simple method is to use turbidity, which is basically how clear or unclear a solution is. So this works by observing a solution of clear nutrients to see if it's going to become more cloudy or turbid as the microbes do grow. You're not growing them yourself, but you're in the fluid and you're not measuring that. You're just kind of looking at it. You can measure it more precisely with things like a spectrophotometer that will measure how much light is able to pass through and how much is reflected back. So another way to measure population size without culturing, if you know how many cells that are in a certain volume of fluid, you can multiply and calculate it by the number in the total volume of the fluid. You can conduct what we call a direct cell count using a microscope. And so this is kind of similar to a white blood cell count. You would have a grid slide or cytometer to count. You can see this here in this picture at right. And so this is going to be very methodical, okay? The limitation of this method is you can't tell the difference between the dead bacteria and the live bacteria. So that's kind of a problem as well. We also have what we call culture counters, and that electronically scans the fluid as it passes through a pipette. A flow cytometer, which works similarly to that, but can measure cell size and differentiate actually between living and dead cells.